when bacteria are put in contact with something like antibiotics, they will evolve, and over time they will develop resistance. And one very good way we have to investigate this phenomenon and ultimately to learn how to prevent it is to reproduce it under controlled and safe laboratory conditions. My name is Marco. I'm a second-year DPhil student in engineering science, and I work on the development of both hardware and theory that can enable us to reproduce and control evolutionary trajectories of microbial populations in the lab. The machine is like a long-term evolution machine where we can basically grow microbial cultures for very long periods of time, and at the same time we can control very precisely the environment in which they grow. We hope that by doing this we can stir the evolution of these bacteria in the way we want, and we can also investigate the way they are evolving with respect to the environment.
Hi, I'm Jess. I'm a second-year DPhil in engineering science. I've been working on trying to engineer a strain of bacteria that will switch on a gene in response to a specific light signal so that we can control them using computers and be able to send light signals to specific cells and activate a response. Using light, we have really fine control over how the bacteria behave, which can help us understand their dynamics and steer them to behave in ways which are useful.
My name is Idris and I'm a postdoctoral research assistant in the Engineered Biotechnology Research Group. Within the DARTS project, I'm developing algorithms, and these algorithms track and characterize the behavior of millions of bacteria. Automating the technology gives us means to make our systems and experiments more accurate. Being able to characterize the behavior of these bacteria would allow us to use them in real-world scenarios. We could eventually engineer plastic-feeding bacteria, but we could also develop new treatments for a variety of diseases.